Welcome to this explainer. Right now, there's actually an invisible, microscopic battlefield active inside your body. And hey, it's not just you. This exact same war is happening inside your dog, your cat, literally every mammal on Earth. Every single second, a highly trained, specialized cellular squad is patrolling your circulatory system. They're hunting down threats and neutralizing them before you even feel a symptom. Today, we're going to meet these microscopic defenders and see exactly how leukocytes function in immunity. Okay, let's dive right into this. We're talking about leukocytes, or as you probably know them better, white blood cells. These guys are the true heroes, the active, living agents of host defense. While a ton of your blood is really just liquid plasma transporting nutrients, leukocytes are the ones out there initiating inflammation, remembering past viruses, and, well, literally eating bacteria alive.
To really appreciate what they do, we kind of have to look at the neighborhood they live in. If you look at a drop of blood, the vast majority of cells are erythrocytes, red blood cells. They're incredibly numerous, they don't even have a nucleus in adult mammals, and they just passively float around carrying oxygen. But our leukocytes? They are way less numerous, structurally complex, and packed with a nucleus and active machinery. They aren't just floating around, they're actively patrolling and doing all the heavy lifting to keep the host alive.
All right, so here's our mission dossier for this explainer. First, we'll map out the mammalian blood battlefield. Then we'll meet the granulocyte frontline defenders, followed by the agranulocyte strategists. After that, we'll see exactly how scientists spy on this cellular squad, and finally, we'll decode the leukocyte pound.
Section 1. The mammalian blood battlefield. Now, what's really interesting about this is that the bloodstream isn't just some dumb pipe. Biologically, it's actually a specialized connective tissue acting as a high-speed highway for our immune agents. Let's do a rapid, high-level briefing of the 5 major special agents traversing that highway. Notice how each cell type basically wears a distinct uniform, specifically the shape of its nucleus. We've got neutrophils with their multi-lobed nuclei for eating bacteria, eosinophils with bilobed nuclei fighting parasites, irregular basophils triggering inflammation, round lymphocytes commanding adaptive immunity, and folded monocytes acting as massive cleanup crews. Let's take a much closer look at that first group. Section 2, granulocyte frontline immune defenders.
So these are our frontline defenders, the ones that rush headfirst into the heat of acute inflammation. They're called granulocytes because their cytoplasm is absolutely packed with these visible granules. But don't be fooled. These aren't just structural anomalies. They are literal weaponized chemical packets. They are loaded with potent inflammatory mediators and antimicrobial enzymes just waiting to be unleashed on an enemy.
The most abundant of these frontline fighters is the neutrophil. If you check out their uniform, they have this highly segmented, multi-lobed nucleus. These guys are the absolute first responders of the innate immune system. Let's say a mammal gets a cut and bacteria get in, boom, the neutrophils rush to the site, guided by chemical signals. Their primary mission? Phagocytosis. That means they are literally engulfing and destroying extracellular bacterial infections in a chaotic, rapid rush of acute inflammation. Next up, we have the eosinophil, which you can spot by a really distinct bilobed nucleus and these prominent reddish-orange granules. Eosinophils have a highly specific tactical role, deploying primarily for parasite defense. So if a dog picks up a parasitic worm infection, eosinophils are the ones attacking it. They also play a huge part in modulating allergic reactions and hypersensitivity. The final granulocyte is the basophil. Now, these are actually the rarest of the white blood cells in peripheral blood, but man, they pack a massive punch. Their granules are so large and dark that they often completely hide their irregular nucleus. When basophils encounter specific triggers, they degranulate, releasing an absolute flood of histamine. This triggers intense inflammation and hypersensitivity reactions. So yeah, they might be rarer, but when they act, the whole body feels it for sure.
Section 3, agranulocyte strategists and cleaners.
Okay, so unlike the chaotic frontline fighters we just met, agranulocytes lack those heavily staining granules. Instead, they rely on strategy and sheer cellular size. First is the lymphocyte. Notice how the dense, round nucleus takes up almost the entire cell, leaving just this tiny rim of pale blue cytoplasm. That massive nucleus, it acts as the tactical command center for adaptive immunity. Lymphocytes basically come in 3 flavors: B cells, which produce antibodies and form immunological memory, T cells, which manage cell-mediated immunity, and natural killer cells, which quietly assassinate virus-infected or abnormal cells. The key takeaway here? They don't just fight, they remember.
The final member of our squad is the monocyte.
And this brilliantly illustrates how dynamic this system is. These are absolutely the largest circulating leukocytes, featuring a really distinct, folded, almost kidney-shaped nucleus. But here is the amazing part. They don't just stay in the blood. They actively deploy. They migrate out of the bloodstream and into the body's tissues. Once they arrive, they transform into massive, powerful macrophages. These are the ultimate cleanup crews that eat cellular debris and actually present pieces of the destroyed pathogens to the lymphocytes so they can plan the next attack.
Section 4. Spying on the Cellular Squad.
Now, to actually observe this invisible battlefield without destroying it requires a pretty meticulous procedure. First, you place a tiny drop of blood on a clean glass slide, and then you use a second spreader slide to smoothly pull the blood across. This creates an incredibly thin film. You have to let it air dry completely, no heat. Then you apply a Romanowski-type stain, like a Wright or Giemsa stain. These specific chemical dyes are exactly what turn those granules red or blue, giving each cell its distinct, identifiable uniform.
Even with a perfect stain, seeing these tiny squads requires immense magnification. Scientists have to use a 100x oil immersion objective lens. And they can't just look anywhere, right? They have to scan what's called the feathered edge to find this perfect, undistorted sector where the red blood cells are evenly distributed and, crucially, not overlapping. It's only in this exact sweet spot that the white blood cells can be properly identified. Section 5. Decoding the leukocyte count. So, how do we decode what we see into actionable clinical intelligence? Well, a scientist manually counts 100 white blood cells, keeping a tally of each specific type they see. This percentage breakdown is known as a differential leukocyte count. For instance, if out of 100 cells, 60 are neutrophils and 25 are lymphocytes, this relative percentage gives us a baseline starting point to understand the balance of power in the blood. But, and this is the crucial point, percentages alone actually aren't enough. I mean, if I tell you 60% of the squad is deployed, that literally means nothing if you don't know the total size of the army. To get real, actionable clinical intel, we have to calculate the absolute count. If a patient has a total white blood cell count of 10,000 cells per microliter and 60% are neutrophils, we just multiply them together. That gives us 6,000 neutrophils per microliter. Now that is a hard, real number we can use. Ultimately, the differential count provides relative percentages, but the absolute count, that reveals the true clinical picture. Relying only on percentages can actually be deeply misleading when you're trying to figure out if a host is healthy or if a specific faction of the immune system is dangerously over or underrepresented. With these absolute numbers in hand, we can really read the intel using specific clinical terms. An overall increase in white blood cells, that's called leukocytosis. If neutrophils specifically are spiking, we call that neutrophilia, and that is a massive, flashing warning sign suggesting active inflammation or a bacterial infection. But here is a huge warning for veterinary immunology. You absolutely cannot use one standard baseline. The normal leukocyte count for a dog is completely different from a horse or a ruminant. You must always use species-specific reference intervals. When we put all of this together, it totally changes how we view the circulatory system. As the source material perfectly states, the blood is not simply a transport system, it is a major pathway through which immune cells detect infection and coordinate immune responses. It really is the central nervous system of our cellular defenses. We've seen the neutrophils rushing to the front line, the lymphocytes commanding from the rear, and the monocytes cleaning up the aftermath. All of it seamlessly orchestrated through the blood. If this delicate, microscopic balance of first responders and strategists dictates the exact line between health and disease, well, it really makes you wonder, what secret battles are the leukocytes in your blood winning right now? Keep learning, keep questioning, and I'll see you in the next explainer.